The first step of recombinant DNA technology is isolation of vector genome as well as your target genome. Today we'll be focusing on the vector genome. Vector is an agent that can carry a DNA fragment into a host cell in which it is capable of replication. We have two different types of vectors, cloning vector and expression vector. Cloning vector is used only for reproducing the DNA fragment or making millions of copies of the DNA fragment or segment of choice, and it is used for creating genomic libraries or preparing probes or in genetic engineering experiments, among other basic studies. Now, expression vectors allow expression of the cloned gene. That is, when you have a DNA fragment, you convert it to an mRNA and then eventually convert it into a protein. That is, express the protein. So usually it will give the product as a protein, and this can be achieved through the use of now the promoters and expression cassettes and regulatory genes, and it is used for the transformation process where we generate transgenic plants, animals, or microbes. For example, you can have a mouse that is cloned with a gene for growth hormone, and therefore once the growth hormone gene is expressed into the growth hormone, that mouse will have a larger size as compared to the normal mouse. So vectors must possess certain minimum qualifications to be an efficient agent for transfer, maintenance, and amplification. And I have a video that I'm going to link up here for the various characteristics that you need to consider when choosing a vector. So vectors are grouped according to nature and sources, and these vectors include bacterioplasmid vectors, we have bacteriophages, we have phagemids, cosmis, viral vectors, animal and plant, shuttle vectors, yeast artificial chromosomes, and transposes. So today we'll be focusing on cloning vectors, then we will have another video on expression vectors. So cloning vectors are a small piece of DNA that you can take from a virus, bacteria, or higher animal that you now artificially make, that can be stably maintained in an organism having been inserted a foreign DNA fragment. So the essence of cloning is that we usually have a foreign DNA fragment or target gene that you have now inserted into a vector so that you can propagate into an organism or host. So all cloning vectors at least usually have certain characteristics. For example, a unique cloning site, that is a sequence that can be cut by restriction enzymes, what we call an MCS. For most organisms, they have a multiple cloning site, so that we have many restriction enzymes that have been grouped together. They also usually have what we call selectable markers like ampicillin or antibiotic resistant genes. So we start off with plasmid. So plasmid vectors are one of the most common vectors that we have. This is the chromosomal DNA within the bacteria. Then we have now what we are calling plasmid DNA. It is circular, double-stranded, and it is extra chromosomal, and it occurs naturally in bacteria and lower eukaryotic cells such as yeast, and they exist in a parasitic or symbiotic relationship with now the host bacterium. Plasmids can replicate autonomously within a host, and they frequently carry genes conferring resistance such as resistance to tetracycline, ampicillin, kanamycin. And the expression of these marker genes are the ones that we use to distinguish between host cells that have been able to carry the vectors and those that do not. We look at PBR322. This is one of the first constructed plasmids that was constructed in the 1977 by Bolivar and Rodriguez. These two people are actually now the derivation of the name BR. So the P stands for plasmid, then BR. And this PBR322 forms the ancestor of so many common plasmid vectors that we have today. It contains an origin of replication because we need to make multiple copies and a gene that helps regulate the number of copies of plasmid DNA within the cell. This origin allows a fairly high copy number. Usually we'll get about 100 copies of plasmid per cell. And this is helpful in cloning because we want to make millions and millions of copies of our target gene. Then it has two markers, ampicillin resistance and tetracycline resistance, and of course has a number of unique restriction enzyme sites that are very useful in now inserting our target gene. So this is how the PBR322 looks like. You can see it has multiple restriction enzyme regions, the tetracycline resistant region, ampicillin resistant region, ORI and ROP. So it has over 40 enzymes, that is unique restriction sites that you can see. For example, we have Icoriv, BAMH1, SPH1, SAL1, SMA3, and DRAL are present within the gene coding for tetracycline. You can see these ones. Then we have others that are within the ampicillin, that is FPST and PVU. And then we have others that are within the beta lactamase, within the beta lactamase gene that provides resistance to ampicillin. And those are the various restriction sites. So when a foreign DNA fragment is inserted into any of these genes, of course, the antibiotic resistance by that particular gene will be lost. If we have the insertion in this area, then resistance against tetracycline, of course, will be lost because this gene has been disrupted. For example, insertion of a restriction fragment in the SAL1 site of the TCR gene, SAL1 here, will inactivate this tetracycline gene. One can still select for the ampicillin colonies and then screen to see the ones that have lost the tetracycline resistance. Bacteriophages or phage vectors. These ones are viruses that specifically infect bacteria. The phage particle attaches to the outer surface of the bacterium and injects its DNA into the cell. The phage DNA is then replicated inside the host and its genes are expressed to make phage capsid proteins, and new phage particles are assembled and released. These phages can accommodate up to 25 kilobases of DNA, and this is more than the plasmids and are often used for preparation of genomic libraries, and they have, of course, a higher transformation efficiency as compared to plasmid. So this is how a bacteriophage looks like. You can see it has a capsid, then the place where we host our DNA. In this case, now we are going to put our recombinant copy. Then we have the sheath, the spikes, and the tail fiber. So they infect bacteria. Some of the examples is lambda phage, M13 phage. So the genome size is about 48,000 base pairs, has high transformation efficiency. It's of course a better version of plasmid vector 1,000 times. It has an origin of replication. Genome is linear within this head. then single-stranded protruding cohesive star ends of the 12 bases, and has also a core site or the site of cleavage of phage DNA. We have what we call phage base, and this is an artificial combination of bacteriophages with plasmids. The positive characteristics of phages and plasmids are utilized. So these are plasmids having a small segment of filamentous phage M13FD or F1 phage capable to carry up to 10 kilobases of DNA. It can be replicated as a plasmid as well as packaged single-stranded DNA in virus particles that can be able now to transfer into a host. So the commonly used phage mid is P blue script 2KS that has been derived from puke 19. Cosmis are plasmids that incorporate a segment of the bacteriophage lambda DNA that has a core site. So cosmids are a combination of now a plasmid that has a core site or cohesive end site, which is called cos in the bacteriophage lambda DNA, and it contains elements required for packaging DNA into lambda particles. It is normally used to clone large DNA fragments that's about 25 to 45 kilobases, and they can replicate as plasmids that is within a suitable host because they have an origin of replication. They can also be packaged into phage capsids so that they are used as an infection tool, that is through the transduction process, and therefore transfer the foreign genes. They have high transformation efficiency, and we've said they can carry up to 45 kilobases. So this is the additional site called the core site from the bacteriophage. They have been used a lot in the construction of genomic libraries. Viral vectors. So we usually have what we call plant and animal viruses that are used to introduce foreign genes into cells for gene amplification. So plant viruses that have often been used are the cauliflower, mosaic viruses, tobacco mosaic viruses, Gemini viruses are 3 groups of viruses that have been used a lot as vectors for cloning DNA into the plant. Then we have animal viruses such as human adenoviruses and retroviruses that are used to transfer genes into cells. So how these ones work is they use viruses that have been weakened that have the capability to infect the host cell. Now in this case can be a plant or an animal. And once the viruses infect the host, they are able to deliver their DNA into the host. For example, we are using human adenoviruses for COVID-19 vaccines, where we are having the use of adenovirus to carry the spike protein gene, and once the human adenovirus infects the human cell, it is able to deliver the DNA or the gene for spike proteins into the human cell. The DNA travels to the nucleus, integrates into the host, and then through the host machinery, it's able to produce proteins that now will be detected by the cell, and the cell is going to elicit or stimulate the immune system. Artificial chromosomes, they resemble the natural chromosomes, but these are synthetic, and these ones are used because they enhance cloning of extremely large DNA fragments, and the eukaryotic artificial chromosomes usually will have telomeres and centromeres. So they can be linear or circular. So they resemble a human chromosome here. This way, it's a human artificial chromosome, and usually you can have 1 or 2 copies per cell. There are different types depending on where the organism they have mimicked: bacterial, yeast, P1 derived, mammalian artificial, and human artificial chromosome. Now the yeast artificial chromosome is used for cloning in yeast. Back and Park are used for bacteria. Mark and Hack used for mammalian and human cells. So they allow extremely large fragments of DNA to be transferred from one organism into another. Transposable genetic elements, we call these jumping genes because they have the ability to jump around from place to place on the chromosomes randomly using what we call enzyme transposes, which will identify inverted repeats, like here, inverted repeats on the ends of the transposon, and once they are detected, they will have cleaving at this area, and this segment of gene will be transposed or will be transferred to another location. So this can be utilized to be able to randomly integrate genes into host cells. We have what we call retrotransposons. Now here is where we have some transposons moving using what we call an RNA intermediate, so that now they will have to first be converted into a complementary DNA by the use of reverse transcriptase, and thereafter be moved to a different section randomly. Let us summarize the target DNA insert size that we need for each of those cloning vectors. This is the vector system, this is the host cell, and this is the insert capacity. So when we are using a plasmid, our host is an E. coli bacterium, and we can have 0.1 to 10 kilobases. Bacteriophages, E. coli again, then we have 10 to 20 kilobase size. Cosmis, E. coli again is the host, 35 to 45 kb. Bacteriophage P1, our host is E. coli, can hold 80 to 100 kilobases. Bacterial artificial chromosome, our host is E. coli, can hold 50 to 300 kilobases. P1 bacteriophage derived AC, the host is E. coli, 100 to 300 kilobases. Yeast artificial chromosome, the host is yeast, 100 to 2000. You can see now that we have a very large fragment of DNA. And then finally we have the human artificial chromosome, whose host is cultured human cells, and it can host over 2000 kilobase pair of DNA insert.